Hi, it's a great pleasure to be here. First of all, I would like to thank the organizing committee for the nice invitation, and it's a pleasure again to collaborate with them. Now at the third Barcelona International Conference on Science and Technology online. So I'm from Lisbon, so my school is engineering school and belongs to the Lisbon Polytechnic Institute. We present diverse bachelor's and master degrees in engineering, including biomedical engineering, where I teach. So as Barcelona, Lisbon is a very nice city to visit. So if you are welcome to visit us. So it's very simple. Go to the Google just to find our internet site. Go to the Google, write these abbreviatures, and you go directly for ESL's webpage. So today I'm going to speak about precision medicine and how it's relevant to discover biomarkers for that and the techniques that we could use for that. So precision medicine means that we want to do diagnosis with more precision for better therapies, for more precise therapies. So using, for instance, the National Institute of Health of the United States and their goals direct for a short period of time and long-term goals. So for short period of time, they are focused as worldwide precision medicine for severe disease as cancer. So characterizing diverse subtypes of cancer will enable this characterization and more precise diagnostic and consequently a more precise therapy. So this is more focus on cancer, but at medium long term, there will be focus on other diseases also. So for that, for instance, at the United States levels, they are considering 1 million volunteers from which they will be collecting biological samples, but also their clinical data and genetical data associated. So the goal is to mechanistically characterize the disease, and for that we're going to achieve a more precise diagnosis and consequently more precise therapies. For instance, if you look at breast cancer, there are very diverse subtypes of breast cancers. Breast cancer is characterized by the overexpression progesterone on estrogen receptors, for instance, a need for more precise medicine therapies that block these receptors, so hormonal therapies. So where cancers that their biomarkers are other kind of receptors, overexpression of other receptors as human epidermal growth factor, receptor, and they need to be treated with, for instance, monoclonal antibodies against these receptors. So if this is already known for breast cancers, along with diverse types of cancers already are starting to be characterized in relation to diverse type of biomarkers that will better define it, enable a better and more precise diagnostic and consequently better therapy. So biomarkers are the main tools towards the precision medicine. So a biomarker is not just a molecule, could be a signature, but should be measurable and quantified. And this, if you measure it, quantify it, related to a defined phenotypic, we could use it for a better diagnostic, prognostic, determine risk factors and therapies. So what kind of techniques we may use to discover biomarkers? Actually, the more efficient are the more recent techniques that enable to analyze many types of molecules, for instance, sequence old genomes as based on next generation sequencing. And this in the last 20 years was amazing, the evolution, as for instance, we achieved from the humanity, we achieved the sequencing of the first human genome in 2003, but after 13 years of research, and more or less $3 billion, so huge investment. And nowadays, after this development, mostly 10 years ago, we are able to sequence a whole genome in 1-2 days and with a couple of 1,000 years, so completely different from the billions. Of course, if you're looking to do a whole set of types of molecules, genes, we are talking about genomics or RNA transcriptomics or proteins and so on. I like to focus on the phenotype because the phenotype is actually what the cell system is really doing. So I like to focus, for instance, in my studies on metabolites. So if we're going to study many metabolites on the system, we are going to study the metabolomes, and the science that study the metabolome and the technique that enables them to achieve the metabolomics. So what kind of technique we can use? Most of the techniques are based on chromatography steps, based on gas chromatography or liquid chromatography, associated at the mass spectrometry to identify the molecules. There's no perfect and ideal technique because each of them will look at a different type of metabolites because all of them have different characteristics as hydrophobicity, as represented here from this slide. Even more, these techniques are highly complex. Not only extracting the metabolome is already complex, the three major techniques besides these two ones based on gas or liquid chromatography associated to mass spectrometry, the third one most used is NMR. All these are highly complex. So in my case, I'm looking to a different technique that is more simply to apply, more economic, more rapid. I've been working, for instance, with Fourier transform in infrared spectroscopy. And just for curiosity, I started to work in bioprocess engineering and apply this technique to monitoring bioprocess. I'm just going to give you some 2-3 examples on this area because I know that normally most of you are from also bioprocess engineer as my background. So for instance, here I applied actually a fiber optical probe working near infrared region of the spectra to a bioreactor containing E. coli that were producing plasmids for gene therapy. And from the single spectra, and we were acquiring in situ in real time, in each 1 minute we have a whole spectra, and we were achieved to retrieve from a unique spectra a set of variables like the quantity of cells inside the bioreactor, the plasmid concentration that was our product inside in cells, and also metabolites as glucose, glycerol, acetate, and so on. And all these models that we developed from a unique spectra that is multiparametric because it enables us to acquire diverse types of information. All these regression models were very accurate to determine these variables as all the correlation coefficients was very near 1 and the error associated was very low, similar to the conventional technique used to detect it, that most of the time was based on HPLC, like plasmids and the metabolites. And we achieved these just for the bioprocess engineer people, based on 2 different setups, one using the fiber optical probe in situ in real time, so acquire online all these variables, all the spectrum and all the spectra we were shift to develop these models, and also at line very rapid in high throughput as based on a microplate with multi-wells. So we achieved this using microorganisms as E. coli, Saccharomyces producing heterologous protein, but also during the expansion of human mesenchymal stem cells. So just determine with high accuracy the glucose, ammonium, lactate, and so on. A very nice information that we could retrieve from the spectra is also the quantities, the relative evolution of some variables as glycogen and so on. How we do that? We could deconvolute it resolve these superimposed peaks based, for instance, on second derivative spectra, as we show it here, and manage this, we could, for instance, determine the levels of glycogen along the period time of our culture and other molecules. So you could metabolic profiling the cells inside the bioreactor. That was our first example, that the technique was very sensitive and accurate to determine the metabolic state of the culture, but also to determine and quantify some metabolites. Concerning the application of the technique for medicine, so we developed after some in vitro experiments. One of them was this one with gastric cell lines that were infected with Helicobacter pylori. Actually, half of the population is infected with this bacteria. That is the major risk factor for gastric cancer, peptic ulcer. So, and this discovery leads to 85 Nobel Prize in physiology. So, looking just so far the spectra of the gastric cells, we have two questions. First, if we manage to detect by the alterations of the molecular profile of the gastric cells if the cells were infected, so, and if you could discriminate slight different kinds of Helicobacter pylori bacteria. And in both cases, the answer was yes. So it was possible to infer if the cells were infected, and discrimination diverse types of bacteria that actually were producing or not different types of toxins. These ones, for instance, are these bacteria that produce these two toxins are more associated to severe diseases as cancer and ulcers. So it was managed to do so. Next, we start to looking in a different kind of study. If we were able, from a drop of blood of a person, to predict the exposure of this individual to genotoxic compounds. On this case, we took around 100 samples from volunteers. Half of the people were professional hospital professionals like pharmacists and nurses that were exposed to genotoxic drugs using chemotherapy. Actually, they were working in hospitals where surfaces were contaminated with these compounds. And half of the other volunteers, the remaining volunteers, were not professionals exposed to these drugs. So for that classically, to evaluate the genotoxic exposure of these persons, blood is taken, the lymphocytes are isolated, they are put in vitro culture, and they are evaluated by another technique, how you call it, cytokinase blocks of the mitosis, to observe chromosomal abnormalities. And indeed, the lymphocytes on the exposed persons, represented in red, in relation to no exposed volunteers, presented chromosomal abnormalities. So we observed at the levels of micronucleus, nuclear buds, and macroplasmatic bridge. So were significantly higher in these hospital professionals. So this is the conventional size, laborious, time-consuming, expensive. So we want to know if taking a single spectra from a drop of blood, we're able to achieve the same results. So we will obtain the spectra from the serum. We pre-processed the spectra by diverse techniques, including the derivatives, and we start to look at the defined regions of the spectra. What we observed was that there was a significant difference between the group exposed in relation to the group non-exposed in different regions of the spectra. That was very nice. Then we look at whole spectra instead of a specific region of the spectra. We also observe, like looking, or just the principal componental analysis, we're just going to start to look at the samples from exposed individual clustering in a different region of the graph on the score plot. Here we have a hierarchical clustering analysis showing that these samples almost also are separated between exposed. So the spectrum is different as a whole from the exposed from non-exposed. Then we developed a model, a classification model, based on support vector machines, and we actually using different pre-processing methods like second derivatives, and we achieve very nice models, enables us to predict from a drop of sample, a drop of blood of a person, the risk associated to the genotoxic exposition of that person. The area under the curve of these models are very high, near 0.9 is very, very high, with high precisions, high sensitivities, high accuracy. So we are able to achieve these results, but in contrary to the conventional assay that takes too long, some days, very complex, we need to take isolate the lymphocytes, maintain the lymphocytes in culture, conduct a cytokinase block assay, and the microscope observation of the nucleus of the cell. So instead of that, we achieve very nice results using the acquisition of a spectrum. This example is a totally different example. This is non-published data, so we are only showing this slide. So looking at the people that received the transplant kidney. So unfortunately, as soon as a person receives a transplant, there is a risk of rejection. So along the years, people were going to lose the organ. So we have actually diverse kind of therapies to reduce the rejection. But of course, if you could predict the rejection process, we could start the therapies earlier as possible. And we want to see if we were able to predict the rejection before, instead of to conduct a biopsy that is very invasive to this organ. And again, we analyze 40 non-transplanted individuals, 30 transplanted individuals with rejections, the other 30 without rejection process. We use more or less 600 serum samples among all these patients, and we are enabled to develop these very accurate prediction models of rejection process before it happens. So before it was detected in the biopsy is amazing result. The last example I bring to you is a completely different technique. So it's infrared, but it's image infrared using a microscope. So this is possible using a special detector. Actually, it's a focal plane array detector. So it has our microscope has a 64 for 64 detectors. So we have 496 spectra acquired using this objective in this area. So in this area of 150 for 150 micrometers, we have 496 whole spectra. That is amazing. So we have prostate tissues from biopsies, and we have a spectrum of each. So if you integrate the finite regions of the spectra, for instance, this is very lipid rich, we're going to reconstruct the distribution of this molecule in these tissue samples in the space, because this is the optical microscopic observation and these reconstructions of this microscopic optical observation in the infrared region. So we are integrating different regions and we are reconstructing the distribution of the molecules on that sample. So in case of the tissue, it's relevant to look at image and not to the average spectra, because in the average we could think we are seeing different spectra. We have here the average spectra of the tissues from presenting hyperplasia and others from adenocarcinoma from prostate. And in average, this look like different, but the standard deviations overlap. So it's not enough to look at the average spectra. So you need to look at the whole picture. So to the image. So to conduct that, we evaluate different preprocessing techniques as the previous one, and multiple scatter correction, that is a physical problem that we need to minimize. Also data dimensional reduction methods, because we have high dimension. So its spectra is 3,600 wave numbers, and we have 4,096 different spectra, and sometimes we don't do just one image. We do 5-10 different images all over the tissue sample. So we evaluate also different classification methods, non-supervised and supervised. Non-supervised doesn't work usually because we have high variability, as usual, associated to human samples, and tissue is more serious. So only the supervised classification as expected was working. So looking at all of these, we achieve very nice, accurate classification in relation to the classification conducted by a physician looking at optical microscopy. Of course, this has many advantages over the human observation because it's free of human error. Like observation usually is associated to high error. Do not depend on the physician's experience, for instance, and is more rapid to detect. So this technique, of course, is not looking just to the metabolome. We are not extracting the metabolites. We are not doing this. So we are looking to the whole picture of the molecules present there, not only for the metabolites, but also proteins, nucleic acids, lipids, carbohydrates. So we are seeing the whole system. So this technique is represents the whole omics. We could say it's very representative of the whole. So in resume, we are working for a precise medicine. So for that, it's very relevant to discover very nice biomarkers defining very specific phenotypes. We could enable us to predict risk factors as genotoxicity, as showing before, or diagnostic as infections, subtypes of bacteria that could alert us for the risk associated also to this patient, but could also be used to diagnostic, as in the case of prostate cancer. So I could also apply this to discriminate different subtypes of cancer. I didn't show that, but spectroscopy is being applied in this case for staging, for example, cancer. Not by our group, but other groups. So it's a very nice technique to discover biomarkers towards a precision medicine. So just before ending, I invite you all to visit us in Lisbon. This is my email, so would be very happy to collaborate with you. So see you soon, hopefully in Lisbon. Bye.